Today.
It's today.
Mm.
That is Wednesday.
Yeah. Now I'm saying Monday.
Okay.
Yeah.
Next Monday, right?
No, that's a exam which will count. Next Monday you will have metabolism exam, which should include amino acids, nucleotides, carbs, and lipids. That's Monday coming up.
Monday or Tuesday?
Monday.
So no
Tuesday we'll go over it because I feel like it was useless to you guys having exam on Tuesday. We'll still do it Monday at the end of the class, and then you go home. After that, and Tuesday, Shukla will go over the answers with you. Okay.
Are we wasting class time?
No, we're not. It's going to be only 20 minutes. 25 questions, 20 minutes. Get it done.
Only a class?
No, the end of the class. 11:30 we'll have the exam. Every 11:30. And that kind of, I want to see who's coming, who's not coming, what you guys doing during the exam, and so forth. I don't know, I feel like I have to be always watching you guys. So, I feel like there's something missing when I don't see you guys take the exam. Honestly, that's why I want to do that Mondays. So he can still go over it on Tuesday with you guys, or the answers, but the exam will be Monday. Now today, the quizzes, it's up to you guys, you want to count it or not, just to see how you do. Just to kind of give you something. It's not necessarily for grades. Exam is for grades. Okay, and I'm not giving you a study guide because it didn't help. It did not help. It's just almost, I feel like, I don't know, I get somehow you guys have the ability to convince me of doing things. You shouldn't be. I feel like giving a study guide is narrowing it down to something small, but even though each question would need the entire knowledge, even the question, let's say it's about cystinuria, but you got to know the entire metabolism of amino acid to answer the question. So by narrowing it down, we're playing the game of high school and undergrad, which is not true here. And it's just almost like cheating you of knowledge. So I don't want to do that. No study guide. Please don't ask me. Can somebody here defend me when they ask me? Because, when you guys ask me, I'm not in the thought. I say, okay, sure, let's do it. Remember, I'm telling you, please don't ask me about study guides. If anybody does, somebody has to hear and remind me. Because if I remember later, I'm going to have to do something crazy then, which is no good. Yeah. It's just not good. It's just not good. You want to be a healthcare provider, you don't look for shortcuts. And it didn't benefit. I'm telling you, it didn't benefit. The scores were not fantastic. It wasn't like somehow you benefited from studying just that aspect of medicine. So, practically it's not benefiting, theoretically it's not benefiting, then why are we wasting our time, At least to me it doesn't make sense. Yes, sir.
Are we going to cover ham and bilirubin?
No, because I said so. Heme and bilirubin is in RBCs. Okay, so we'll discuss that in the following terms when we're talking about red blood cells, circulation, hemodynamics, cardiology, and things like that. They design it that way, doesn't mean, it's the right way. So biochemistry, everything is taught from all over the place. For example, I'm not going to talk about biochemistry of chloride channel right now, because it's not right time. It's not time. We'll discuss it in the respiratory system. We're not going to talk about biochemistry and pharmacology of beta blockers, ACE inhibitors right now. It's not time. So, okay. All right. It is metabolism. So, again, metabolism is related to GI. So, I want to finish. This is what you should be doing right now. So, since metabolism, there's two things in metabolism. How do you get proteins? Okay. Carbs, nucleic acid, and lipids into cells. Now when I say cells, it means all cells. Okay? And then what happens within the cell? Biochemistry, so far what we're doing is we're doing what happens within the cell. Once we finish that, we'll come back to this part, which is the digestion part. Digestion. Okay, which is the digestion part. So as I said, it's difficult. I'm trying to make it organized in a way that will make more sense for you. Okay. But these books, they will organize it the way my book. You know, if there's a mistake, you have to remind me, because it's not edited. I'm the author and I'm the editor. Could I have made mistakes? Of course. You know, I took my notes and gave it to students like, please type this for me. That's all. And put in this fashion, and I put it. It took me actually all the books to put all my notes over a month, because I was forced for the ASMLE to do these. This wasn't even for this school, it was for the ASMLE program. So, but I haven't had time to edit them yet or anything. There are mistakes in them. The only book you trust is hardcore medical books, like Guyton, Robbins Pathology. BRS's I wouldn't. There's still mistakes in it, because it's a board review series. It's different. These books, they're good, concise books, but they might have mistakes. So when you read something and then conflicts with what I say, always check with me. Always check with me. Either I'm wrong or they're wrong. There's nothing in between, right? And also internet. Be careful. You know, like you read something on the internet, you think it's right, and later on you find out it's not wrong, not right. Not everybody has the ability to read complicated scientific issues and explain it or write it. They don't read the whole story, or they read it and misunderstand it. There's a lot of details to it. So, and then if you just read superficially, they say, you know what, this inhibits that. But yeah, there's more to it. If it stimulates something else, it will inhibit that, maybe, you know. So always check with those books. If you have something, if you are gonna search online, Medscape, e-medicine is the best source in my opinion. E-medicine. It's a lot of reading, but guess what? It's better than reading Wikipedia or this or that 20 different websites. Just stick to one, and it has a lot of great information, and it's authentic, and it's edited, and it shouldn't have mistakes. If they do, it's minimum. You know, everybody's gonna have mistakes. It's impossible not to, because we're a human being. We make mistakes. The way we perceive things sometimes. Use glucose to do so. It's metabolism. So think about what I'm saying is, and it's a simple term, but it can get you confused. Metabolism means the sum of all chemical reaction in your bodies. Chemical reaction can be any reaction. Sometimes you break things, sometimes you build things. You want to divide in two categories: catabolism and anabolism. Okay, so any change in physiological chemical structures is metabolism in a sense. Okay, so we're only going to talk about right now what happens intracellularly. I'm only talking about what happens intracellularly, not how you bring these macromolecules from the environment after digestion. We're talking about after digestion, but there's something that happens here too. We'll talk about GI. There's breakdown, there's redistribution, there's accumulation, and then there's of course secretion, and all of that we'll talk about it. But now to understand metabolism, we have to understand the heart core of metabolism, which is the mitochondria. Cellular metabolism depends on what? Mitochondria. Although you've been told what mitochondria is the source of ATP, or it's the powerhouse of the cell. That's all you know. But if you really think about it, entire metabolism comes back to what? ATP. The whole idea of metabolism is ATP. How do you get that ATP, right? So mitochondria is the center of metabolism. Mitochondria is the center of metabolism. Cells that don't have mitochondria, that have no nucleus, cannot be considered living in the long term, and they die fast because they're not able to generate what? ATP. And if you remember, I said that how do you define life? Somebody said depends how you look at it. I'm not talking about life in the social perspective. How would you define living things? Maybe that's more accurate. How do you define something that's living? One of the criteria is they must be able to generate ATP or energy. That means mitochondria is essential. Okay, mitochondria is essential for higher organism that needs more ATP. For simple single cell organism like bacteria, RBCs, and some other single cell organisms like protozoa, yeah, you don't have to have mitochondria. If you do, it's not that developed, or they might have only one. Bacteria don't have mitochondria, but they're still living because they're able to generate ATP by glycolysis or fermentation or so forth. But nonetheless, there has to be a mechanism. For eukaryotic cells or humans, mitochondria is the most important organelle amongst the cell. Okay? So, and you know that we talked about necrosis, we talked about hypoxia. One of the major sign of permanent damage is what? Lethal damage, irreversible cellular damage, is mitochondrial damage. Mitochondrial damage. So it's essential. Either when the chromosomes are destroyed, DNA. See, without mitochondria you cannot live long enough. But without nucleus you can live, you might not be able to repair. So at the end of the day, there are some organelles that are absolutely critical for survival, and mitochondria is the first one. Okay? So whenever we talk about all of these metabolism and so forth, please focus on mitochondria. I know a friend of mine, if he's listening right now, he loved mitochondria so much he called himself Mitochondria, his last name or nickname. When he figured out there's so much to it, although it's a simple organism. Anyhow, let's go to carbs.
Is that real?
Huh? It is real. He just called him his if you hear me, listen to me. His Isna, you're not supposed to sit in the back. You're supposed to sit somewhere close so I can see you.
Unless I my spot.
No, his email was mitochondria something, and he would call himself for the nickname. So I thought it was amazing. But anyhow, okay, he will not be able to survive in higher altitude at all. Okay, let's start with carbs. Okay, so when we talk about carbs, any sugar product is called carbs. Any sugar product is called carbs. Okay. So in terms of carbs, we will talk about 3 different types of carbs: monosaccharides, disaccharides, and poly. Okay. Monosaccharide is the single unit, the simplest unit of sugar. What is the basic simple unit of proteins? Great job. Therefore, what is the simple single unit of carbs? Monosaccharide. Don't say glucose, although it's a major one, but there's more. Just like how many types of amino acid do you have? 20. When I say what's the building block of protein, you don't say glycine, right? You don't say that. You say amino acid. So when you say what's the building block of sugars, say monosaccharide. Don't say glucose, fructose, or galactose, or something else. Okay? Alright, so there's 3 monosaccharides that clinically that are important. Clinically that are important, and at least in medicine we have to know them. Maybe there's more, we're not concerned with them. Fructose, glucose, and one comes from far away galaxy. One comes from fruit. Galactose. Okay. Okay. And then some people do believe that, milk comes from a far away galaxy. Yeah, so galactose comes from milk. But anyways, I don't believe that. Okay, so just to be on the record. Fructose. The source of fructose is primarily fruit. Fruit, not vegetables. Fruit. Especially orange, apple, bananas, things like that.
How about the onion?
Onion, is it a fruit? How do you define fruit?
Seeds.
Anything that has seed. Onion has no seed. So yeah, tomatoes have it. Garlic has seeds? Then it has it. Some fruit have them more than others. Just like some of you are more inclined to become surgeons or doctors than the rest of the humanity. So just have that flexibility. When I say fruit, does not mean every fruit will have 100% fructose. Although honey is rich in fructose. Honey. Okay. So a good source of. Now, when you're eating about fructose, just to get your interest, the fastest source of ATP. The fastest source of ATP is fructose. C6, HO6. So look, 1 carbon, 2, 1, 2, 3, 4, 5. 6. All sugars look like that. Okay. 6 carbon. Now each carbon should have how many OHs?
2.
2. But always here there's an OH. You see that the hydrogens are not usually written, it's assumed that it's there. Everybody should know from organic chemistry, when you have a carbon, it has 4 potential bonds available, or unvalent or unpaired electron or valence electron. So if 2 carbons are involved in this bond, you will see the corner like this, corner of a sugar. And then that means there's 2 available. One of them is always OH and the other one is H. So that means each carbon has how many H's?
2.
One here and one attached to the oxygen. So you just have to know that. The difference with the sugar comes, honestly, I don't care for it. It's just to kind of get you interested and kept going. We don't care for recognizing glucose versus fructose versus galactose based on shape. It's not our job. Nobody's going to say, oh, if somebody has galactosemia, fructosemia, oh, what was the shape of the sugar? Nobody cares for that. Okay? So that's for organic chemists and so forth. If you went to UIC or whatever other universities and you were taught that, it's good to know, but you know, we don't care for it. It doesn't help. Maybe help your question once in a million time. But anyhow, the way we differentiate between fructose, glucose, and galactose, all of them are the same structure, but depends on which way is the OH pointing. Let's say for glucose, I think it's for fructose, I don't remember exactly, or for galactose, all the OHs are pointing down. They're all pointing down. But let's say in some cases one is pointing up. Immediately it's a different shape. Receptors react to it differently. Enzyme reacts to it differently. Channels react to it differently because they're different shape. Different shape. They're not the same. Okay? It's like comparing, you know, apple and oranges. So they look different. That's how they're different. Okay. But anyhow. So what do we need to know? The source of fructose is fruit. The source of glucose is vegetables, especially sugarcane, potatoes, and stuff like that. Okay. The source of galactose, milk. So minimum you have to know the sources of them. Because if somebody becomes intolerant, it would help you to identify them. What are they eating to cause this intolerance in people? Fruit, fructose, F for fructose, F for fruit. Okay. Glucose, canes, no, googers, sugars. Okay, so vegetables and energy, or ATP. Glucose is primarily used for ATP or energy. ATP or energy. Primarily. It's not only for energy, it's primarily for energy. Let me tell you something. About 80% of your blood glucose, 80% of your blood glucose is used by the brain to make energy out of it. Huge. So that means the organ in your body that depends on glucose is who? The brain. So whenever you have hypoglycemia, think that your brain is starving. Okay? But that's not the only source for brain. They can use ketones. But glucose is the primary source. Fructose is the primary source of ATP for sperm. You need to know that. Sperm does not like glucose, does not like galactose, likes fructose. Okay? If you get confused, think of sperm liking female and sperm likes fructose, both F's. Okay? Or fertilization, fructose. Okay? Whatever correlation you can make to remember. Yeah?
Does the brain use only glucose or also fructose?
They could, but they'll be converted to glucose. Yeah. So look, we talked about fructose, we talked about glucose. Glucose is the primary source of ATP for brain and skeletal muscle. Especially brain. 80% of your total plasma glucose used by who? By brain. That's a big deal. That's one thing. Now when it comes to galactose, galactose is used as an antigen on the surface of cells. Antigens, recognition, ID, passport, driver license. So galactose can be attached to the surface of cells in different shapes and formats. and it becomes an ID card for your cells. So you know these are your cells. For example, when you talk about RBCs, they have antigen A, type B, type C, type O, not C. I almost went as far as C, but that's half a O. It's okay. So know that those antigens have galactose attached to them. Also, galactose is used in brain development. Specific lipids have galactose attached to them, or glucose attached to them. So children who are born with galactose metabolism defect could have mental retardation. Okay, are we good with these three monosaccharides? So know their shape, generally speaking. Overall, they're hexagon. Okay, I think fructose, when they're open, they'll all be in this shape. OH, 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 and depending which way it's pointing out, it'll be different sugar. Okay, and all sugar should open up in solution. When you find them in water, they should open up, and then they can be used. If you want to store them, you don't open them. If you want to use them, you break them, then you open the ring. Okay. Now let's move to disaccharides. Disaccharide, we have sucrose, we have lactose, and we have maltose. Okay. Sucrose come from sugar canes, vegetables, and fruit. Okay. Lactose come from milk. Maltose primarily comes from potatoes and things like that. Like most. So if I were to say the same thing about vegetables, sucrose comes from fruit, maltose from vegetables, lactose from milk. Okay. Now, there is a big problem we have. Since we're not talking about digestive system yet, but I'll tell you something quick. Lactose, disaccharides are too big to be absorbed in the GI, in your intestine. They're too big. You have to break them into monosaccharides. The reason we're focused on monosaccharides is because they're the only sugar molecule that can easily cross in and out of the cell. Lactose is too big. It's two sugars, two monosaccharides. Sucrose is big and maltose is too big. So they have enzymes to break them. Now, sucrose is made of what? Let's just do one thing. All three of them are their first sugar. It doesn't have to be the first, but at least one of the sugar is glucose. All three of them have glucose. They're disaccharide. If they're disaccharide, that means they're made of two sugar molecules. They're not mono, they're di. All three of them, at least one of their sugar molecules should be glucose. Okay, and that's how we differentiate them, is based on the second one. Again, it's not necessarily, the order is not important, okay, so it depends how you look at it from left to right. Okay, so sucrose will be glucose and fructose. Very good. Fructose. Lactose should be glucose and galactose. Glucose and glucose will give you maltose. Okay. Remember, this is what makes them different. Okay. But if they're too big, now you heard of lactose intolerance, right? The problem is you cannot break down lactose. If you cannot break down lactose, lactose will accumulate in the intestine because it cannot be reabsorbed. If you cannot absorb it, it will stay in the intestine. It's too big. The only thing you can absorb in the intestine is what? Monosaccharides, monosaccharides, monosaccharides. Monosaccharides are small enough, and we have transporter for monosaccharides. We'll discuss them in the GI. Okay, but just to let you know, lactose intolerance because you cannot break down glucose and galactose into two independent free sugar molecules. So you cannot break down lactose. And lactose accumulates in the intestine. And if it does so, that means it retains water. If it retains water, stool never becomes solid. It stays dilute, diarrhea. When lactose reaches the colon, bacteria there. You have bacteria that ferments lactose, like E. coli, and so forth. And these bacteria will break down lactose into lactic acid and carbon dioxide. So now the carbon dioxide will cause distension of the intestine, and that will cause pain. The only pain receptor you have in the intestine are stretch receptor. If you have too much gas, what happens to the intestine? It stretches, becomes balloon, and it hurts. So pain, diarrhea, bloating are all the symptoms of what? Lactose intolerance. It's just simply because you can't break down lactose. Okay. But now that means we should have enzyme. The enzyme that breaks glucose and fructose, what would you call it? Sucrase. Whatever you want to break down. If you're breaking down sucrose, the enzyme is sucrase. If you're breaking down lactose, the enzyme is lactase, maltase. 3 enzymes, and these enzymes are primarily found in the jejunum, okay, and duodenum. Okay, so a small intestine, let's say. Okay, and they're brush borer enzyme. Write it down. They're called brush borer enzymes. They're called brush borer enzyme. They're found on the surface of enterocytes or villi and microvilli. They're called brush borer enzyme. I'll slow down. Brush borer enzymes. So the enzyme that breaks these bonds are known as brush borer enzymes. Is that okay? They're found in the small intestine. Small intestine. Small intestine. Very good. Located in the small intestine. Okay. Now, small intestine have these cells, and these cells are known as, the entire GI has them. They're known as enterocyte. Enterocyte. Enterocytes. Gastroenterology. Gastroenterology. Entero means intestine. Okay. Enterocytes, they have this finger-like projection from one of their surface, the luminal surface, and then they will have these tiny little things sticking out. They're known as villi and microvilli. Villi and microvilli. Villi are finger-like projections. Microvilli are even tiny finger-like projection sticking out from villi itself. Each villi will have multiple microvilli. And this is just simply to increase surface area. Surface area. The best example would be, like you've seen Christmas trees, right? Those little pellets that stick out from the main branch, the tiny green ones. Sucrose comes in contact. Let's say, for example, as sucrose comes in contact with it, it breaks it down into glucose and fructose, and then they are absorbed. So they're attached on the surface of. Now, why is that important? What if you're eating crazy food? What if you're eating some sort of crazy food, and this food causes erosion of these brush borer enzyme? Could it lead to problem, lactose intolerance? Yes. What if you have viral infection, which causes death of enterocytes? Could it decrease the number of these enzymes? Yes. What if you drink alcohol? What if you're drinking, let's say, dry cleaners, oven cleaners? People do crazy things. What if people are just like in love with eating mud? There are a lot of stuff that are toxic, so anything that's toxic can cause erosion of this. But the thing is, your GI is able to replace these cells more frequently. But if you have a toxin that will cause damage much faster, then you could have lactose intolerance. Okay, lactose intolerance. Alright, so we're good with this. So you know the 3 disaccharides, know their name. Know their name. Now remember, lactose is digested, broken down in the intestine. But lactose is made in breast. Lactose is made in breast. Okay? So that's a different game now. Babies, they need milk. Breasts are able to make lactose by combining what? Galactose and glucose. Okay? It's too big to be absorbed. And we'll discuss the enzyme beta-galactosidase later on. Polysaccharide. Polysaccharide, when you talk about that, means many sugar molecules combined together. Many. Not one, not two, not three. Many. A huge chain. Huge chain. A chain of gold, like how what's those singer guys? They wear it. Cool. So huge chain. Now, this chain is called polysaccharide. You can call it monosaccharide. 2 will be called? 3? Trisaccharide. 4? Tetrasaccharide. 5? Penta. Keep going, and then eventually you get tired. You're like, let's call them polysaccharide. Okay, so, but we are not going to go talk about tetrasaccharide, saccharide, trisaccharide, and we're not going to talk about penta and hepta and deca and so forth. Let's just say 3 is enough. Okay. Polysaccharide, we have the part that is polysaccharide in our body is known as glycogen. Polysaccharide in our body is known as glycogen. Glycogen. Now, there are 2 types of glycogen in our body. One is hepatic. The other one is muscle. Okay, so we have 2 sources, 2 types of glycogen. Now, when I say muscle glycogen, you must make this conclusion that muscle uses glucose. Muscle uses glucose for energy. Otherwise, why would muscle want to have its own glycogen? You have to make that conclusion. I don't have to always tell you. Okay. The primary source of ATP, or the preferred fuel of the rest of the body, is fatty acid. Keep that in mind. Write it down somewhere so you keep this in mind. The primary source of ATP or energy by the body is fatty acid. The exception is the brain. Okay. The rest of the body, especially internal organs, prefers, even your heart loves fatty acid. Skeletal muscle loves fatty acid. Okay. But it also uses glucose, especially when you work out. Okay. Alright, so now muscle glycogen is for what? For muscle ATP only. It's only to deliver ATP or make ATP for muscles. That's all. The only thing you can convert muscle glycogen to is what? ATP. Okay? You can only convert this glucose stored in muscle, this polysaccharide stored in muscle, for ATP. It has no other functionality. Keep that in mind. Later on we'll talk about it when you reach gluconeogenesis. Hepatic glycogen, on the other hand, primary function of hepatic glycogen is to keep blood glucose level normal, prevent it from going down. If you have a store and you have low glucose blood level, what are you going to do? You're going to break that store. You can take glucose from that store and bring it to blood. But the reason I'm saying brain is because 80% of blood glucose is used by what? By the brain. Okay? So look, the primary function of hepatic glycogen is what? To restore blood glucose level. To maintain blood glucose level normal, within normal range. What's normal glucose level in blood? Very good. Okay, 100 is the best option. Just 100% glucose, 100 milligrams per deciliter, something like that, I think. Right? No, milligram per deciliter. Liter. Why did I say letter? Liter. Okay. So we want to maintain blood glucose level within that range. When it's high, hepatic glycogen will be stored. When it comes less than 80, when it becomes 60, let's say, when blood glucose level drops to 60 or less than 80, then you will have to break down hepatic glycogen. As we speak, how do you get glucose? How do you get glucose? Diet, of course. I don't see anybody having some special magical source. You have to eat. Now, when you eat, how much do you eat? I hope you have changed that one third. If you haven't done, I hope you have done that, but you know it's only for your good. Now, when you're getting that glucose, your body uses glucose continuously, non-stop, correct? Because from your liver, never muscle. Please, muscle is only for muscle. Yeah, muscle is only for muscle. For brain is only hepatic glycogen. Never confuse the two. Muscle glycogen is only for muscle contraction, only muscle contraction. Hepatic glycogen is to restore blood glucose level. And blood will take this glucose where? Where? So look, blood glucose level is for all organ, for the entire body. So hepatic glycogen is for the entire body. The reason I'm putting brain, because I earlier told you 80% of your total blood glucose is used by the brain. So it's the primary source. I mean consumer, sorry. Okay, so it's okay. It is true that hepatic glycogen is for the entire body to be used. But 80% of that is used by who? By the brain. So in essence, the only function that hepatic glycogen serves is to increase blood glucose level, or prevent it from going down, or keep it at the same range. That's all. It shouldn't increase above normal. It should maintain it between 80 and 100. That's the purpose of hepatic glycogen. It's a store glucose. And what would you store glucose for? When you have less, you use that. You had a question? Yes?
Do you want to make sure so by default for the muscle glycogen is only 20%, or is that not straight?
Oh, you mean so this hepatic glycogen that we break down, if blood glucose 80% is used by the brain, so the other 20 goes to muscle, is that what you're saying?
Right.
No.
No, not from the hepatic, just the muscle being produced, the muscle glycogen.
Mhm.
Is that only 20% then?
No, this is nothing with plasma.
It's just for the muscle itself.
Look, so when you eat carbs, you will store glucose in muscle as glycogen and in liver as glycogen. They're two different stores.
Okay, that has
Nothing to do with each other. Yeah. This is a bag in the basement and this is a bag in your kitchen. You have nothing to do with it. So you say this one is only for using muscles, but it's not one is more than other. No, that's not true. Yeah. Once you break down your hepatic glycogen, and then blood glucose level, majority of the blood glucose level goes to the brain. 80% of your total blood glucose level is used by the brain. The only time you can store is if you have extra. By then, consumption is not a problem. The only time you can store glucose is after eating, after a meal. And that's when you would need insulin. Okay, are we okay with this? So you have two sources of glycogen. One is hepatic, one is muscle. Good? Muscle glycogen is only for muscle use. Hepatic is for blood glucose level. That's all. But majority of the blood glucose is used by the brain. And then the second majority is used by the muscle itself. So even this will go back to muscle in the time of workout, exercise, or stress, or things like that. Okay, and then minority is used by other organs. Other organs they love to use fatty acid. Fatty acid is a main source of ATP. Fatty acid. Okay. All right. So at least we know these molecules. I mean these type of sugars. The last thing I want to do is before we go, we have some hormones that have to do with the metabolism. Okay.
Dr. Munia,
Yeah.
the only source of glucose is just carbs. So that means if I eat proteins all the time, I will get no glucose in my body.
You will get it, but with a lot of difficulty. Okay, this is why I'll answer your question, then we'll take a break. But I feel bad for your kidneys because they will be under a lot of stress. When you only use one diet, they become under a lot of stress. And let's say amino acid. And especially which amino acid is mostly that we use to this? All of them could be. I'll just use amino acid, that's fine. Amino acid. And here I should have said monosaccharide. So you can include fructose and galactose as well. You see that they can be converted to what? To acetyl-CoA. Okay? Now we have nucleotides. They could be converted to acetyl-CoA. The opposite is also true. Acetyl-CoA could be converted to amino acid, could be stored as fatty acid, could be made into glucose. So if you only eat amino acid, that's converted to acetyl-CoA. If you need glucose, it'll be converted to glucose. It's just not a very good process. It's more expensive. It puts a lot of stress on your body. Okay? So it doesn't matter. You can eat nucleotide, you will have all three of them, but it takes time. If it's used, the body is not used to this thing. The body is used to having all four sources of nutrition.
So where do I get the nucleotides?
No. You can get protein and look. Do you remember when we talked about nucleotide synthesis? Aspartate, glutamine, glycine are converted to what? Nucleotides. Okay. The sugar component comes from sugar. The nitrogen base comes from amino acids. The phosphate comes from phosphorus. So yeah, if your diet is deficient in one of these things and you're eating everything else, your body is able to compensate for it. There's interconversion occurring between them all the time. When you eat pure amino acids or pure fatty acid or pure glucose or pure nucleotide, it's a problem. When you eat a steak, for example, steak has amino acid, has nucleotides, has fatty acid, small amount of glucose, if any. It has all four of them. Most dietary products have everything in them. Some have more protein, some have more nucleotide. For example, wine has more nucleotide, red meat has more nucleotide. Okay, cheese and eggs and things like that have more amino acids, especially tuna and stuff. And then lipid product have more, or fatty product has more fatty acid, and vegetables and fruit have more carbs. So yeah, it's true that some have more of the other. At the end of the day, everything, that's why you should eat everything. Okay, but it doesn't even matter. You know, like people when they diet, they think, let me erase this. Now when we have a central molecule, we can have a different chart. That nucleotide can be converted to this, and this can be converted to that, and this can be converted to that, and this can be converted to that, and this and this, and this and this. Glucose can be converted to one of these. So and it's never plausible based on these evidence that, you know, when you diet or you do this, or I'll eat this, it wouldn't happen. No, as long as you eat too much amino acid, what happens? You will use them. So let me just give you quickly. Now let's say you're only eating protein. Protein is converted to what? Amino acids, right? Okay. So you will use whatever amino acid to make enough ATP your body can function. And you still have extra amino acid. What's going to happen to this extra amino acid? If you're working out, it might be stored as proteins. You'll increase your muscle mass, muscle size, and other things. And on top of that, you're not satisfied. You're eating even more amino acid, even if you work out. If you don't work out, then it'll be stored as, so some of them will be converted to as glucose, and glucose will be stored as glycogen. So I'll escape that step. Just say glycogen. By now you know enough. So it'll be stored as glycogen. It could be stored as, let's say, triglyceride. Okay. Now, triglyceride, by the mechanism of our body, is known as permanent stores. Okay. Glycogen is temporary store. Okay. ATP, if you need it, of course you need it. Now protein is not a permanent store or not a temporary store. It is primarily for the functionality of the body. So the only time, if you're eating amino acid, it will be converted to protein if you work out. If you work out. If you don't work out, you're not working out, you're just eating, then it will be glycogen. But temporary stores are small. They're small. They don't have the ability to stretch and store as much as you want. Their purpose is not permanent store. So when they're filled up, what happens to the extra amino acid? Will be converted to triglyceride, which is the storage form of lipids, or we call fat by our definition. So then it doesn't matter. You can eat all amino acid you want. If you don't use it to make energy, if you don't work out, then of course it's going to be stored as these two. All the extra will be stored as fat. Okay. So and that goes for fatty acid. Permanent store. And this is only if you work out and do things. Otherwise it will not help. Protein, you have to have the need for protein synthesis. Okay. Let's change this from glucose to fat. Let's say you eat fat, fatty acid. Fatty acid, again, it's preferred, step one, to be used as energy. But how much energy can you use? Depends how much you're walking, how much you're thinking, how much you're working, how much you're doing things. It all depends on your activity. And it's not only your physical activity. Majority of activity comes from the brain. Okay. And if you have enough ATP, you just made enough ATP, your body says, I have enough, that's enough. It's just like when you make enough money, the rest you're going to invest or store in banks. So the rest will be stored as temporary. But of course, fatty acids are easier to store as triglyceride compared to glycogen. So that's a disadvantage of eating pure fat. Okay, because it's easier to store as a permanent fat. Whereas glucose have to be converted to fat. Protein have to be converted to fat to store it permanently. So it doesn't really matter. It all comes to one thing and one thing. How much are you acquiring, and how much are you consuming, and how much are you using? If you're consuming more than using, it would accumulate. If the balance is exactly the same, nothing would change. If you're consuming less and using more, you will starve. So there's only three things at the end of the day. It depends. I mean, it's just, your body is a perfect businessman. Yeah, let's take a break.